Assalamualaikum warahmatullahi wabarakatuh. So today our presentation is all about rapid antigen and molecular tests for viral infections.
Viral infectious disease is one of the main concerns of health organizations as it can cause global pandemics such as COVID-19 that happened during 2020. Thousands of lives have been taken because late detection and treatment. Conventional method less valid as it are not suitable for a pandemic situation. As the technology grows, many modern diagnostic tools have been developed such as rapid antigen and molecular tests. There are some differences between conventional and modern method of diagnosing viral infections. Firstly, turnaround time for conventional method take days to weeks just to produce results compared to modern method that only take minutes to hours. Secondly, conventional method have low sensitivity to high sensitivity based on what method is used. Unlike modern method, they have moderate to high sensitivity. Thirdly, for costing, modern method cost low to moderate while conventional method are high in cost and it does require specific equipment. Fourthly, high-skilled technician and specialized laboratory include in training requirements for conventional method. In contrast, modern method were invented as friendly user. And lastly, conventional method directly detect virus while modern method detect viral protein or genetic material of the viruses.
And now I will proceed to rapid antigen tests. The principle of rapid antigen test is to detect antigen based on finding a particular immunogenic component, typically protein or polysaccharide, that is a part of the pathogen or other biological entity. The immunoassay detection of specific antigen that detects host-produced antibody triggers by an antigen help in diagnosis of infectious disease, non-infectious autoimmune, and inflammatory disorder. So, how lateral flow immunoassay work? A lateral flow immunoassay works by the act of capillary to move a liquid sample through a paper-based strip that contains a specific biochemical that will react with specific antigen or specific antibody. Then, the sample pad is where the liquid sample such as blood, urine and saliva were applied. The pad helps prepare an optimal flow and filter out large particles like red blood cell. Then, the conjugate pad is where the sample flow and contain dry detection reagent, usually antibody or antigen attached to nanoparticles such as colloidal gold which produce red line. Nitrocellulose membrane is the reaction area and have the test line T line and control line C line. At the test line, the gold labeled antibody will recognize an epitope, a unique part of the target, and binding occurs. And the last part, which is the absorbent pad, is located at the end of the strip and is helped to maintain a consistent fluid flow across the nitrocellulose membrane by absorbing excess liquid and prevent a backflow. Latest innovation in rapid antigen detection system. So firstly, it enhances sensitivity and accuracy, since the test is more sensitive and providing accurate result with new mechanisms like optical detection technology and chemiluminescence. Then, using digital readers and AI to interpret results can reduce human errors and provide a qualitative analysis result. Next, a multiplex testing, which is the ability to test for multiple pathogens simultaneously, such as polio, COVID-19, and influenza A and B home kit test. Lastly, alternative detection mechanism, an advanced platform which can be 10 to 1,000 times more sensitive than a conventional antigen test. For an example, like CRISPR versus the system. Stages and disadvantages of rapid antigen test. The advantages is antigen test is cheaper and fast. Then, it's also a useful public health tool due to a rapid result that likely accurate and help identifying highly infectious person in a community. Then, it also a potential for use in point of care setting where we can rely on the rapid nature of antigen assay. The disadvantage of rapid antigen test is it has a lower sensitivity than PCR test, and this could lead to false negative result. Then, we have less expert interpretation in point of care setting that could lead to less professional healthcare workers' interpretations of a result. And lastly, laboratory space, supplies and staff are still required. Just because antigen testing can be done at home doesn't mean that most antigen tests can be done outside a certified laboratory without a proper training.
Hello everyone, so today I will briefly explain about how molecular diagnostic works and some of the latest innovations in this field. First, the principle is very easy. The virus is detected by its genetic material, whether it's RNA or DNA. Since the virus may be present in very small amount, then we usually use amplification testing such as RT-PCR, LAMP, CRISPR-based detections to make million copies of the target and then make it very detectable. The most common method is RT-PCR, so it is widely used in detecting COVID-19, dengue, and influenza. It involves three major steps, which are denaturation, annealing, and also extension. Then it is followed by analyzing whether the viral RNA is present. The procedure involves sample collection. expression, RNA extraction, reverse transcription, amplification, and finally detection. Today, new innovations are making molecular testing very accessible and fast. The first one will be point of care system, so it includes Gene Expert and also IRDINA, which provides results in minutes using a sealed cartridge. Second, multiplex panels, such as BioFire. It tests for many pathogens at once, so it helps to guide for treatment very quickly. The third one, CRISPR-based diagnostic, offers in highly specific and rapid detection. And lastly, RT-LAMP provides a very fast result without the use of complex machine. These technologies are widely used in diagnosing COVID-19, dengue, influenza RSV, where rapid and accurate detections are very essential in early treatment and also proper patient management.
So now, let's compare these diagnostic tools side by side. So, rapid antigen test is fast, 15 to 30 minutes only, and very cheap, around 5 to 20 ringgit. But sensitivity is only 60 to 80%. Lab-based molecular tests, however, highly accurate at over 95%, but take 4 to 6 hours and cost 100 to 300 ringgit. So, the breakthrough is point of care molecular that is combined both, which over 95% accuracy and in just 15 to 45 minutes. Examples include Gene Expert and IRDINA. But when do we use which test? So, we use rapid antigen for mass screening, symptomatic patient, and home testing when speed matters. So, use molecular tests for confirmation, early detection, and high-risk cases when accuracy is very critical. So, these tools complement each other, not replace it. So how about the real-world impact? So in Malaysia, rapid tests were integrated into MySejahtera, and NS1 antigen tests help manage 130,000 dengue cases yearly. So hospital use multiplex panels for respiratory infection. How about the global? Globally, enable pandemic control, safe travel, and a school reopening. So about the clinical benefit include faster treatment decisions, better infection control, and improved patient outcomes. So for the future, multiplex testing, smartphone integration, and sub-10-minute molecular test is in development. So to conclude, these tools revolutionize viral diagnostic. Rapid antigen provides speed and access. Molecular provides accuracy. So point of care molecular offers both. So innovation continues with multiplex panels and CRISPR based tests. So this innovation permanently change how we diagnose and manage viral infections worldwide. So thank you for your attention. That's all from us. Thank you.